Welcome to NeuroCurio. In this series, we explore everyday topics through the lens of neurology. I am Fable, and I establish the subject.
I am Vera, and I report the object.
Today, we will consider the leaf not as flora, but as a blueprint that medicine has learned to read. In neurology, its pattern appears in the whorled patterns on skin, an exquisite tree-like branching of our brain's neurons. By examining this delicate plant appendage, we trace a unifying story of form, lineage, and connection.
First, ash leaf macules in tuberous sclerosis. Tuberous sclerosis arises from loss of function mutations in the TSC1 or TSC2 genes, which encode a protein complex that inhibits the mechanistic target of rapamycin mTOR signaling pathway. mTOR dysregulation leads to uncontrolled cell growth and multi-organ hamartoma formation. In the skin, melanocyte development and migration is disrupted, producing hypopigmented, leaf-shaped macules in over 90% of affected individuals. In the central nervous system, cortical tubers, subendemnal nodules, and subendemnal giant cell astrocytomas develop, contributing to high rates of epilepsy, intellectual disability, and autism spectrum disorder. Cardiac rhabdomyomas, pulmonary lymphangioliomomatosis, and renal angiomyolipomas can occur. Second, hypomelanosis of Ito. Somatic mosaicism involving chromosomal or monogenic variants affects both skin and neural tissues during embryogenesis. Disrupted melanocyte function and melanin synthesis results in streaks of hypopigmentation, with leaf-like patterns following Blaschko lines, lines of normal cell development in the skin. Neurological associations include intellectual disability, developmental delay, abnormal muscle tone, seizures, and occasionally hemimegalencephaly. Third, the leaf-like Purkinje cell, primary output neuron of the cerebellar cortex, which has a distinct branching morphology that resembles a tree. Pathologists studying this cell have likened the axon to the roots of the tree, the cell body as the trunk, and the extensive dendrites as the leaves and branches. In Purkinje cell degeneration such as in spinocerebellar ataxia, cerebellar circuitry is disrupted, causing progressive gait ataxia, limb incoordination, dysarthria, nystagmus, and oculomotor abnormalities, with symptom severity reflecting the degree of cell loss.
These associations lend us to two important concepts. Melanocytes and neural tissues share a common embryological origin from the neural crest.
This underpins the reason for the existence of many neurocutaneous syndromes, including tuberous sclerosis and hypomelanosis of Ito.
Neurons resemble trees visually.
There are shared features of hierarchical branching, spatial territory occupation, and connectivity patterns. Neuronal arbors, like tree branches, extend and bifurcate to maximize coverage and connectivity within a given space, and their density and territory size follow predictable relationships important for neural computation.